Hello there, and welcome to Fall Asleep. Tonight, we are going somewhere most people will never see. Not a forest, not a mountain at dawn, not even a coral reef where sunlight still filters through warm water. Tonight, we are going somewhere far deeper, far darker, and far stranger. We are going to the deep ocean floor, to a place where the seafloor does something that should not be possible, where the ocean holds a lake inside itself, a body of water sitting inside another body of water, separated by a boundary as still and clear as the surface of any lake on a calm summer morning. These are called brine pools, and they are among the strangest places on this planet. They sit in the deep dark, far below where sunlight reaches, far below where most life is expected. They are still, almost perfectly still, filled with water so dense with salt that it does not mix with the ocean above it. It simply holds its boundary. Patient, quiet, ancient, a shimmering golden line marking the edge between two worlds that exist in the same ocean, on the same seafloor, in the same dark, and yet are separated as completely as if by glass. This is not a legend. It is real. It has been filmed, studied, and sampled by remotely operated vehicles descending through thousands of meters of open ocean to reach it. And what scientists have found there has changed our understanding of where life can exist and what life can endure. Before we descend together into the quiet dark, if you find these journeys restful, I invite you to like, subscribe, or leave a gentle thought below. It helps other tired minds find their way here, one sleepy soul at a time. But for now, there is nothing you need to do. Let your jaw unclench. Let your shoulders drop. Let your breathing slow into something easy and natural, like the breathing of someone who has already let the day go. And as your mind begins to gently release the weight of everything it has carried today, join me as we sink slowly downward, into the blue, into the black, into the still and extraordinary world of the brine pool. Let us begin. Tonight is not the first time humans have wondered what lies at the bottom of the sea. For centuries, the deep ocean was the great unknown. Too dark to see into, too deep to reach, too cold and too pressurized for any diver to survive. The first serious attempts to measure the depth of the ocean happened only in the 19th century, using weighted ropes lowered by hand from the deck of a ship. It was slow work, imprecise. It gave only a rough sense of the depth at a single point, nothing more. It was not until the mid-20th century that sonar technology allowed scientists to map the seafloor systematically, and not until the development of deep-sea submersibles, vehicles capable of descending thousands of meters under enormous pressure, that anyone could actually see what was down there. What they found surprised everyone. The deep ocean was not a barren, flat desert. It was a world of ridges and canyons, of volcanic vents and cold seeps, of brine pools and communities of life that no one had predicted. It is a world we are still learning to read. To understand a brine pool, we first need to understand something about salt. Salt is not simply a flavoring. It is not just something shaken over food at a table without much thought. Salt is one of the most powerful physical substances in the ocean. It governs buoyancy. It governs density. It decides which water sinks and which water rises, which currents flow where, and which parts of the ocean become layered into bands that barely mix even over centuries. Ordinary seawater contains roughly 35 grams of salt per liter. That is about 3.5%. It is enough to make seawater denser than fresh water. It is enough to let us float more easily in the ocean than in a swimming pool. But brine is not ordinary seawater. Brine is water saturated with salt far beyond what ordinary seawater contains. In a brine pool, the salinity can be 3 to 8 times the salinity of normal seawater. In some extreme cases, even higher. Water so thick with dissolved salt that it behaves almost like a different substance entirely. So dense that ordinary seawater simply cannot mix with it. So heavy that it sinks to the seafloor and stays there, held in place by nothing more than the physics of density. No container, no barrier, no walls. Just the fact that it is too heavy to be lifted by the water above it. When brine and ocean water meet at their boundary, they do not swirl together. They meet. And they hold. The boundary between them is sharp and shimmering. In underwater footage, it catches the light from a submersible's lamps in a way that looks golden, or silver, or like heat rising off summer asphalt on a still afternoon. This is an optical effect caused by the difference in density and refractive index between the two water types. Refractive index is how much light bends when passing from one medium into another. At the halocline, light scatters differently above and below. The result, to the camera, is that shimmer. That golden line, that impossible seeming surface within a surface. The brine below, the ocean above, and between them, that quiet, shimmering boundary, still and patient, in the deep dark of the seafloor. There is a word for the boundary layer between the brine and the overlying ocean. Scientists call it the halocline. Halo from the Greek for salt, and cline from the Greek for slope or gradient. A salt gradient, a place where salinity changes sharply over a very short vertical distance. Haloclines exist in many parts of the ocean, wherever fresh water meets salt water, wherever surface conditions differ significantly from the water below. But nowhere are they as sharp, as dramatic, or as stable as at the boundary of a deep brine pool. Most haloclines in the open ocean are transitional zones, fuzzy gradients where salinity changes gradually over meters of depth. Brine pool haloclines are different. They are boundaries, sharp enough to look like a surface, sharp enough to film, sharp enough that a submersible crossing it produces footage that looks like descending through the surface of a lake, even though you are already kilometers below the sea surface. The physics that creates and maintains this boundary is the same physics that keeps oil floating on water. Two fluids of different density simply do not mix readily. The denser fluid settles below, the lighter fluid sits above, and the line between them holds, patient and precise, for as long as the difference in density remains. But where does brine come from? Why does water this salty exist at all on the ocean floor? The answer is written in the ancient geology of the seafloor itself. Hundreds of millions of years ago, parts of what are now the ocean floor were not ocean floor at all. They were shallow inland seas, cut off from the larger ocean. Lagoons, salt flats, enclosed basins sitting under the hot sun of ancient climates that no longer exist, on a planet whose geography we would barely recognize. Over millions of years, those shallow seas evaporated. The water lifted away into the atmosphere, carried by winds across a world unrecognizable to us. But the salt remained. Salt does not evaporate. It stays behind, accumulating, concentrating, building up into thick layers of halite and other evaporite minerals as the water that carried them rose away and vanished. These ancient salt deposits became buried under later sediments. Layer upon layer of silt, sand, and ocean mud settled on top of them over millions of years, pressing down, compressing, covering the old salt with newer material until it lay deep below the seafloor. And there the salt sat, locked in stone, patient, until something disturbed it. In geologically active areas, the weight of overlying sediment creates enormous pressure on buried salt layers. Salt is not rigid the way rock is. Under pressure, it behaves almost like a slow fluid. It flows, it rises, it pushes upward through the sediment above, following weaknesses in the rock, moving with the strange slow patience of something that has nowhere to be but eventually arrives. This process is called salt tectonics, or halokinesis, and when those ancient salt deposits finally reach the seafloor, they dissolve. Seawater seeping down through cracks contacts the old salt and dissolves it. The water becomes saturated, dense, heavy, too heavy to rise, too heavy to be carried away. It pools, it accumulates, it forms a lake on the seafloor, within the ocean, held in place by density alone. The exact age of a brine pool is difficult to measure, but geochemists can analyze sediment layers that have accumulated over the pool, layer by layer, century by century, undisturbed. Reading those layers gives researchers a record of ancient currents, ancient chemistry, and ancient climate, written in the mud beneath an underwater lake that most of the world does not know exists. The timescales involved in the formation of brine pools are difficult to hold in mind. The salt that feeds many brine pools was deposited during the Mesozoic era. When the world was warm, shallow seas covered large parts of the continental interiors, and the ancestors of today's mammals were small and cautious. That salt was buried under sediment during the slow accumulation of millions of years of ocean floor material. It has been sitting underground, under pressure, waiting, for longer than the Atlantic Ocean has existed in its current form. This is what the deep ocean deals in. Not years, not centuries, not even millennia, but deep time. The kind of time that makes human history seem like a brief flicker at the end of a very long story. And it is in deep time that brine pools operate. Their chemistry is ancient. Their biology is ancient. Their sediment records are ancient. They are places where the very deep past reaches up into the present, where the ocean floor offers a glimpse of a world that existed long before anything alive today had evolved. The Gulf of Mexico is one of the best studied locations for brine pools on Earth. The Orca Basin, discovered in the 1970s, sits at roughly 2,300 meters depth. It is a depression in the seafloor, roughly 30 kilometers long and 20 kilometers wide. And within it, brine sits like a filling in a bowl, dense, still, cold, separated from the overlying ocean by that sharp, shimmering halocline, the boundary layer where the two water types meet and refuse to blend. When the first scientific expeditions sampled the Orca Basin, the results were striking. The brine was almost entirely anoxic, devoid of oxygen, a place where most of the life processes we associate with a healthy ocean could not function. Aerobic bacteria that would normally decompose organic material could not survive. Fish could not breathe. The brine seemed, in this sense, like a dead zone. And yet, even there, even in that dense, cold, oxygen-depleted water, life had found a way. Microbial communities, adapted to extreme salinity and near-absent oxygen, lived within the brine itself. Not at its surface, not at its edges, inside it. The deep seafloor surrounding brine pools in the Gulf of Mexico has also revealed extraordinary communities, organisms that derive their energy not from sunlight, but from chemical reactions happening where brine meets sediment, where methane seeps upward from below, where sulfur compounds cycle in the dark chemistry of the deep. Mussels the size of a palm, clustered at the edge of the brine, tube worms anchored in the sediment, extending feathery plumes into the water above, shrimp grazing across pale bacterial mats, and beyond them, the archaea. These are microorganisms from one of the three great domains of life, older than bacteria in their lineage. At the edges of brine pools, certain archaea perform a process called anaerobic oxidation of methane. They consume methane seeping upward and use it as their energy source, working in partnership with sulfate-reducing bacteria. Together, these two groups consume enormous quantities of methane before it can reach the water column above. They are a natural methane filter for the deep ocean, performing a quiet service in the dark, with no fanfare, by organisms that were old when the dinosaurs were young. The communities found at brine pool edges in the Gulf of Mexico are not uniform. Different pools and different edges of the same pool support different assemblages of organisms. The chemical conditions vary from place to place. Some areas are richer in methane. Others are dominated by hydrogen sulfide. Some have higher oxygen concentrations in the overlying water. Others are more fully anoxic. Each microenvironment within the larger brine pool landscape selects for a different set of organisms, a different combination of metabolic strategies, a different food web. This patchiness makes brine pool ecosystems unusually rich in their overall biodiversity. Across the entire brine pool landscape, more species are represented than any single location within it would suggest. It is another version of the edge effect, the tendency for biological diversity to increase at boundaries and transitions. The Gulf of Mexico seafloor, scattered with brine pools, cold seeps, and methane hydrate deposits, is not a uniform habitat. It is a mosaic, a patchwork of microenvironments, each with its own chemistry, its own community, its own quiet story unfolding in the dark. Let us slow down here and think about what it means for life to exist without sunlight. For most of human history, sunlight was considered the fundamental requirement for life, the essential ingredient. All food chains, all ecosystems, all the green and growing world traced back to the sun. Photosynthesis is the process by which plants and algae capture light and convert it into chemical energy, into sugars, into the foundation of the food web. Every animal that has ever lived on the surface of this planet ultimately depends on sunlight through this chain. Remove the sun, and the food web collapses. This seemed obvious. It seemed certain. Then, in 1977, a research submersible called Alvin descended to the seafloor near the Galapagos Islands, in the eastern Pacific Ocean, 2.5 kilometers down. No sunlight, no photosynthesis, no connection to the surface world at all. And what the scientists found changed biology. Around hydrothermal vents on the ocean floor, in water heated to extreme temperatures by volcanic activity below, life was flourishing. Tube worms 2 meters tall, anchored to the basalt. White crabs moving across the rock. Dense clusters of mussels, bacterial mats covering everything. An entire ecosystem, dense and thriving, in total darkness. It ran on chemosynthesis. Bacteria at the base of this food chain used hydrogen sulfide, pouring from the vents, as their energy source. They converted chemical energy directly into organic matter, without light, without photosynthesis, without any connection to the world above. They were the foundation. Everything else ate them, or ate the things that ate them. Sunlight was not needed. Not even slightly. This discovery changed the definition of the conditions necessary for life, and brine pools, studied in the years that followed, deepened that change further. Even without the heat of vents, even without sulfur-laden hydrothermal water, life persisted at the cold, still, chemical boundaries of brine. The rules of life turned out to be far less strict than anyone had imagined. The universe of possible homes for living things turned out to be far larger. The implications of chemosynthesis extended well beyond biology. They reached into geology, into planetary science, and into the philosophy of science itself. Before 1977, the search for life elsewhere in the solar system was heavily focused on Mars, because Mars was the most Earth-like planet in terms of surface conditions. It had a thin atmosphere. It had a history of liquid water on its surface. It was at a distance from the sun that seemed at least marginally compatible with life as we understood it. After 1977, that focus broadened dramatically. Scientists began to look at the moons of Jupiter and Saturn with new eyes. These were worlds that received almost no sunlight. From the perspective of photosynthesis-dependent life, they were hopeless. But from the perspective of chemosynthesis-dependent life, they were interesting. They had liquid water beneath their icy surfaces. They had geological activity. They had chemistry. They had, in other words, the things that matter, rather than the things that had always been assumed to matter. The discovery of chemosynthetic ecosystems did not just expand the list of places life might exist. It changed the definition of what it means for a place to be habitable. There is a particular creature associated with brine pool environments that is worth pausing on. The Halophiles methanicola. A polychaete worm, small, pink, roughly the length of a thumbnail, unremarkable in appearance to a casual glance, the kind of creature that might be overlooked entirely by someone scanning a seafloor image for more dramatic subjects. But its habitat is among the most extreme any animal on Earth inhabits. This worm lives directly on methane hydrate, a substance found at the edges of some deep brine pools on the Gulf of Mexico floor. Methane hydrate is sometimes called methane ice or fire ice. It is a crystalline solid in which methane molecules are trapped inside cages formed by water molecules arranged into a lattice of ice. It only forms under very specific conditions of high pressure and low temperature, conditions found on the deep ocean floor but essentially nowhere in our surface world. On the surface, methane hydrate is unstable. Brought up too quickly from the deep, it fizzes and dissipates, releasing methane gas into the air. But on the seafloor, under the weight of kilometers of water, it sits there solidly. White, crystalline, dense, almost beautiful in the beam of a submersible's light. And on the surface of this methane ice, Halophiles methanicola makes its home. It burrows into it. It grazes on the bacterial communities that colonize the hydrate surface. It exists in an environment that has almost no oxygen, that is saturated with methane, that sits under pressures that would kill any unprotected human instantly. And it lives there, quietly, going about its small life in the deep dark, completely undisturbed by the apparent impossibility of its situation. At some cold seep environments nearby, researchers have also found vestimentiferan tube worms growing on the sediment. These grow far more slowly than vent tube worms, just a few millimeters per year. But they live far longer. Some cold seep tube worm colonies have been estimated at 200 years old or more, living quietly in the dark, attached to the sediment, century after century, without moving, without leaving, without any apparent need for a different life. It is a reminder that the strangest places on Earth often turn out to be some of the most inhabited. The methane worm and the cold seep tube worms are part of a broader category of organisms that scientists call extremophiles, from the Latin extremis, meaning extreme, and the Greek philos, meaning loving. Lovers of the extreme. The study of extremophiles is one of the most rapidly growing fields in biology. Every time researchers explore a new extreme environment, they find organisms that have found ways to make a living there. Extremophiles have been found in the boiling water of Yellowstone hot springs, in the briny waters of Antarctic ice lakes, in the highly acidic runoff of abandoned mines, in the dark interior of salt crystals thousands of meters underground, and in the highly pressurized, pitch-black sediments of the deep ocean floor. Each new discovery pushes the known boundaries of life further. Each new discovery makes the universe seem a little more accommodating. Not all extremes are the same. Some organisms are adapted to high temperature, others to low temperature, others to high pressure, high acidity, high salinity, or high radiation. Some organisms, called polyextremophiles, can tolerate multiple extremes simultaneously. The Halophiles methanicola worm, living in anoxic, high-pressure, methane-saturated brine pool edges, is one such organism. Life, it turns out, is extraordinarily creative in finding ways to persist. The Red Sea contains some of the world's most extraordinary brine pools. This narrow sea wedged between northeastern Africa and the Arabian Peninsula is one of the youngest ocean basins on Earth. It is, geologically speaking, a new ocean in the making. The African and Arabian tectonic plates are slowly pulling apart, spreading the seafloor, widening the basin by roughly 1 to 2 centimeters per year. In 10 million years, the Red Sea may be a full ocean. Beneath its deep central basins, ancient salt deposits from evaporating seas lie buried under sediment, and in the deepest parts, at depths between 2,000 and 3,000 meters, those buried salts have created a collection of brine pools unlike anywhere else on Earth. Some of the Red Sea brine pools are hot, not slightly warm, hot. The Atlantis II Deep, named after the research vessel that discovered it in 1965, contains brine at temperatures exceeding 60 degrees Celsius, and with salinity roughly 7 to 8 times that of normal seawater. In the deep ocean, where background water temperatures are typically close to 2 degrees Celsius, 60 degrees is remarkable. The heat comes from hydrothermal activity below, from the same geological processes slowly pulling the plates apart, even here, in water that is simultaneously extremely salty, extremely hot, and nearly devoid of oxygen. Microbial life has been found at the boundary layer, where cooler and more oxygenated water meets the dense, hot brine. Communities of microorganisms thrive. Anaerobic archaea, from one of the oldest lineages of life on Earth, have been recovered from the boundary layers of Red Sea brine pools. They are cycling nitrogen and sulfur, producing methane, contributing to global biochemical cycles, happening invisibly, in the deep dark, at the bottom of a narrow inland sea. The sediment beneath these pools also holds something remarkable. In anoxic brine, organic material is preserved with unusual fidelity. Pollen from ancient land plants, carried by winds across the desert. Microscopic shells from marine organisms of different eras. Organic compounds from biological activity in the waters above. All of it preserved in the undisturbed mud. The Red Sea brine pools are not just biological curiosities. They are geological archives, records of ancient climate, written in sediment, held in stillness, in the deep dark. The discovery of the Atlantis II Deep in 1965 was, at the time, genuinely shocking to the scientific community. The research vessel Atlantis II was conducting a routine oceanographic survey of the Red Sea when its instruments detected an anomaly in the water temperature readings at depth. The water was far warmer than it should have been. When samples were brought up, they confirmed what the instruments suggested: a body of extremely hot, extremely salty water, unlike anything previously described in the ocean. The discovery triggered a wave of scientific interest in the deep Red Sea. Further surveys revealed that the Atlantis II Deep was not alone. Other hot brine pools, known as deep brine depressions, were scattered across the central rift valley of the Red Sea, each one different in temperature, salinity, and chemistry. Together they formed a landscape of extreme environments, unlike anything found elsewhere in the world ocean at the time. The scientific community initially struggled to explain them. The connection to the buried evaporite salt deposits and to the geological spreading of the Red Sea floor took years of work to establish. But once established, it provided a model that researchers could apply to other parts of the world ocean, eventually leading to the discovery of brine pools in the Gulf of Mexico, the Mediterranean, and elsewhere. The Eastern Mediterranean holds another remarkable collection of brine pools, discovered more recently than those in the Gulf of Mexico or the Red Sea. They have produced some of the most striking footage and scientific findings of any deep-sea environment on Earth. The Medee brine pool, discovered in 2011, sits in the eastern Mediterranean at a depth of roughly 3,500 meters. It is one of the largest brine pools known anywhere on Earth. Its surface area is estimated at roughly 1 square kilometer. Think about that for a moment. A lake 1 kilometer across, sitting on the floor of the Mediterranean, 3.5 kilometers below the surface, undisturbed by the shipping lanes above it, undisturbed by the storms that cross that busy sea. And its boundary layer, that golden halocline, is so sharp and so visually dramatic that footage of submersibles crossing it looks almost like descending through a liquid mirror. Light scatters differently above it. Light scatters differently below. The colors change. The texture of the water changes. The two water types meet with a clarity that still surprises scientists who have spent entire careers studying the deep ocean. The eastern Mediterranean sits over ancient geology shaped by the collision of the African and Eurasian plates, by the closing of ancient seaways, and by the Messinian salinity crisis, a period roughly 5 to 6 million years ago when the Mediterranean nearly dried up entirely. Vast quantities of evaporite salt were deposited during that drying, when the Atlantic reconnected with the Mediterranean through what is now the Strait of Gibraltar. The sea flooded back in what geologists believe was one of the most dramatic flooding events in Earth's history. The salt remains buried, patient, slowly dissolving upward into the brine pools of the deep Mediterranean floor. What makes the Medee pool particularly interesting is not just its size. It is what lives at its edge. The halocline turns out to be a zone of intense biological activity. Organisms here can take advantage of both worlds simultaneously. The oxygen from above, the concentrated chemical nutrients from the brine below. Certain microorganisms have evolved to live precisely in that narrow band. Not in the ocean above, not in the brine below. At the line between them, in the golden shimmer itself. A home built at the edge. The footage of the Medee brine pool crossing has been seen by millions of people online. It is one of those genuinely astonishing pieces of video, the kind that makes people stop and watch a second time, and then a third, simply because it seems impossible. A submersible descends through clear, dark ocean water, and then it crosses a surface. A real, liquid surface. Below it, everything looks different, slightly hazier, slightly different in color. The submersible's lights reflect differently. The camera seems to be looking through two different substances, because it is. That footage does something important that scientific papers alone cannot always do. It makes the abstract concrete. It takes a physical phenomenon described in equations and measurement units and converts it into something the eye can grasp immediately. There it is, an underwater lake in the ocean. Visible, real. Researchers who have spent years working on brine pools in the laboratory, analyzing water samples and geochemical data, have described watching that footage for the first time as a genuinely affecting experience. To know something exists and to see it are different things. The Medee footage collapses that difference. It is why remotely operated vehicles, with their cameras and their lights, have been so important to the study of the deep ocean. They do not just gather data, they show us the world. Let us think for a moment about what happens when an animal falls into a brine pool. This is not a hypothetical. It has been observed. Footage from remotely operated vehicles has captured the edges of some brine pools scattered with the remains of creatures that entered and did not leave. The brine is so dense that an animal cannot simply sink into it. A fish approaching the pool would encounter the halocline. The dense brine below would resist its weight. The fish would rest on the surface of the brine as if lying on a liquid floor. Held up by density, floating on water inside water. It is a strange and striking image. But the brine itself is not a comfortable place to rest. The extreme salinity causes osmotic shock. The body of the fish contains cellular fluid at roughly normal seawater concentration. But suddenly it encounters water far saltier than its own tissues. Water moves by osmosis from areas of lower salt concentration to areas of higher concentration. From the fish's body fluids outward into the surrounding brine. The fish dehydrates rapidly. Its cells lose water. Its tissues shut down. In minutes or less, it is dead. But then something unusual happens. The brine preserves it. The absence of oxygen, the extreme salinity, and the cold temperature of the deep ocean all combine to slow decomposition dramatically. Bacteria that would normally break down a dead animal cannot function in the anoxic brine. Some brine pool edges have been found to contain remarkably well-preserved remains of animals that fell in perhaps years or decades before, preserved far more effectively than most deliberate preservation methods could manage. The brine pool does not consume what falls into it. It keeps it still, cold, preserved. In the quiet dark, far from the world above, researchers have noted that organic material preserved in brine can retain molecular detail for extraordinary lengths of time in anoxic, cold, hypersaline conditions. The rate of degradation slows dramatically. Some researchers have suggested that brine sediments may preserve chemical fragments from organisms that lived in the waters above thousands of years ago. A record of who lived there, written in molecules, preserved in mud, waiting for instruments sensitive enough to read it. The deep brine is patient. It holds what it is given. The preservation properties of brine have been known, in a practical sense, for a very long time. Before modern refrigeration, salt was one of the primary methods humans used to preserve food. Meat packed in salt, or stored in brine solutions, resists bacterial decomposition far longer than unsalted meat. Fish salted in barrels could survive long sea voyages. Vegetables brined in salt water could be stored through winter. The mechanism is osmosis. In a salty environment, bacteria cannot maintain the internal water balance necessary for their cellular processes. They dehydrate and die. The same mechanism that preserved food in kitchens and cellars for centuries operates, on a vast scale, in the anoxic brine of the deep ocean floor. The difference is that in the deep ocean, the preservation is even more complete. Low temperature slows chemical reactions. The absence of oxygen eliminates aerobic decomposers entirely. The extreme pressure suppresses many biological processes. Together, these conditions create a preservation environment more effective than anything a kitchen or a laboratory could produce by design. It is one of the many places where the deep ocean turns out to have been doing something practical and useful in its own way, long before humans thought to look. There is a category of organisms that does not merely live near brine pools or at their edges. These organisms live inside the brine, not at the boundary, not at the fringes, inside the densest, saltiest, most chemically hostile part of the pool itself. They are called halophiles, from the Greek halas meaning salt and philos meaning loving. Salt lovers. True halophiles not only tolerate extreme salinity, they require it. Remove the salt, reduce the concentration to something an ordinary organism would find comfortable, and the halophile cannot function. Its enzymes are adapted to work specifically in high salt conditions. In normal seawater, those same enzymes would misfold. They would lose their shape. They would fail to catalyze the reactions the organism depends on. Its cellular membranes are structured to resist the osmotic pressure that would destroy a normal cell. Its entire biochemistry has been tuned, over enormous spans of evolutionary time, to operate in conditions that represent the extreme end of what dissolved water can contain. Extreme halophiles are found in some of the saltiest environments on Earth. The Dead Sea, which has a salinity roughly 10 times that of normal seawater. The Great Salt Lake in Utah. Salt evaporation ponds around the world, where the concentrating water turns vivid pink and orange with the density of halophilic populations. In the Dead Sea, most organisms cannot survive, but halophilic archaea flourish there. They color the water in dense blooms of pink and orange, their pigments serving as protection against intense ultraviolet radiation. The domain archaea was not recognized as distinct from bacteria until the 1970s, when Carl Weiss and George Fox used molecular techniques to compare the genetic sequences of microorganisms from very different environments. What they found was startling. Certain organisms, the methanogens, the extreme halophiles, the thermophiles of hot springs, were as different from bacteria as bacteria are from animals. They belonged to a separate domain of life, equal in fundamental importance to bacteria and to all plants, animals, and fungi. This discovery reorganized the tree of life, and it revealed that the deep brine pools of the ocean were not biological backwaters. They were windows into the deepest past. The discovery that archaea represented a fundamentally separate domain of life had profound implications for how we understand the history of life on Earth. Before Weiss and Fox's work in the 1970s, life was divided into two categories: prokaryotes, organisms without a cell nucleus, which included all bacteria, and eukaryotes, organisms with a cell nucleus, which included all plants, animals, fungi, and single-celled organisms like amoebas. The new three-domain system, with bacteria, archaea, and eukaryotes, as three separate and equally fundamental branches, changed that picture entirely. It also raised a striking possibility. If archaea are as deeply different from bacteria as bacteria are from eukaryotes, then they may have diverged very early in the history of life. They may represent a lineage that separated from the common ancestor of all life at a very ancient moment. Some researchers have proposed that the common ancestor of archaea and eukaryotes was itself an archaeon, meaning that we, as eukaryotes, are more closely related to the halophilic archaea of the brine pools than either of us is to bacteria. The creatures thriving in the densest, saltiest, most inhospitable water on the ocean floor may be our very distant relatives. Quiet, ancient, persisting in the dark. One of the quieter aspects of brine pools is the role they may play in the story of life itself. Not just as a home for extreme organisms today, but as a possible stage for the very first chemistry that led to living things. The origin of life is one of the deepest unsolved questions in all of science. We know that life began on Earth somewhere between 3.5 and 4 billion years ago. We know the earliest life forms were microbial. We know life was present in the oceans before it appeared on land. But the specific environment in which the first living systems assembled themselves from non-living chemistry remains genuinely unknown. Several candidate environments have been proposed: hydrothermal vents on the deep sea floor, hot springs on land like those in Yellowstone, shallow tidal pools where chemistry could concentrate as water evaporated, and deep-sea cold seeps and brine environments. Brine pools offer some chemically interesting properties. The extreme salinity concentrates organic molecules. In dilute ocean water, the chemical building blocks of life, amino acids, lipids, nucleotides, the precursors to proteins and genetic material, drift far apart. In concentrated brine, they are packed close together. Interactions that might be vanishingly rare in open water become more probable in the dense chemical environment of a brine pool. The sharp halocline provides a natural interface where chemistry from below meets chemistry from above. And interfaces are where interesting chemistry happens. Membranes form at interfaces. Concentration gradients develop at interfaces. Energy flows across interfaces. One of the central puzzles of early life chemistry is explaining how the first cell membranes formed, how life separated itself from its environment and became a distinct enclosed thing. The mineral surfaces and sediments surrounding brine pools also matter here. Clay minerals have layered structures with large surface areas. Organic molecules adsorb onto those surfaces, concentrating in ways that might facilitate reactions needed to build longer, more complex molecules. Metal sulfides can catalyze the reduction of carbon dioxide into organic molecules under the right conditions. At a brine pool edge, concentrated chemistry, mineral surfaces, and a sharp physical gradient all come together. Not proof of anything, but an interesting convergence of conditions that origin of life researchers find worth considering. One of the most striking features of brine pool science is how recently most of it has happened. The Orca Basin was described scientifically in the 1970s. The hot brine pools of the Red Sea were discovered in the 1960s. But the Medee pool was found only in 2011. And there is every reason to believe that many more brine pools remain undiscovered. The deep ocean floor is vast. Our ability to survey it in detail is limited by the cost and difficulty of deep-sea expeditions. The remotely operated vehicles that allow us to film and sample these environments are expensive to build and operate. The ships that carry them require large research budgets. As a result, the deep ocean remains the least explored large environment on Earth. We have mapped the surface of Mars at higher resolution than we have mapped the floor of the ocean. Entire brine pool landscapes may exist in understudied parts of the Pacific, the Indian Ocean, or the deep Arctic, simply because no one has yet sent a vehicle to look. Each expedition that returns from the deep ocean brings back discoveries that revise our understanding of what is down there. The brine pools we know about are almost certainly a small fraction of all the brine pools that exist. You are lying very still now. The day has grown distant. The thoughts that pressed close earlier, the plans, the conversations, the small worries that felt large, have begun to soften at their edges. Your breathing has found its own pace. Slow, easy, unhurried. And as you rest here, imagine descending. Not quickly, slowly, the way a single leaf falls through still water. Without effort. Without effort. Without rushing. Simply following where gravity and stillness lead. The surface of the ocean is far above you now. The light that once filtered down in pale shifting columns has faded to a deep blue, then a faint gray, then nothing. Here there is only dark and quiet. A quiet unlike anything on land. No wind, no traffic, no insects at dusk, no distant murmur of voices or machines. The deep ocean is one of the quietest places on the planet. Not entirely silent. There is the low, distant clicking of deep-diving whales passing through the water column kilometers away. There is the barely perceptible settling of soft sediment. But these sounds are so different from anything familiar that the overall effect is profound quiet. Something that asks nothing of you. The seafloor appears below, pale and soft in the beam of a submersible's light. Undisturbed sediment that may not have been disturbed in thousands of years. And ahead of you, something catches the light differently. A shimmer, not a reflection of rock. Not a trick of sediment. Something liquid that bends the light with a quality unlike the water you have been descending through. You are approaching a brine pool. The boundary comes into view. It looks like a still lake surface seen from above. But you are already deep underwater. There should be no lake surface here. And yet there it is. That golden shimmer. That impossible line. At the edges of the brine, where it meets the sediment, white bacterial mats spread across the seafloor. Dense and pale, soft and slow, and the pool itself stretches out in perfect stillness. A lake within an ocean. In the deep dark. In the quiet. The submersible hovers just above the halocline. The pilot is careful. Moving too quickly through the boundary layer creates turbulence that can temporarily disturb the halocline, churning the two water types together briefly before gravity reasserts itself and the boundary reforms. This is one of the strange properties of the brine pool. It is remarkably stable, but not entirely inert. Disturbances at the boundary are self-correcting. The physics of density differences quickly restores the separation. This is different from many other boundaries in nature, which, once disrupted, take time to rebuild. The halocline reforms on timescales of minutes after a disturbance. Not hours. Not days. The density difference is so large, the restoring force so strong, that it reasserts itself almost immediately. There is something almost alive seeming about this, although it is entirely physics. A boundary that, when disturbed, reasserts itself. A line that refuses to be erased. That property has made brine pools more durable than they might otherwise appear. Research submersibles have crossed haloclines hundreds of times in the course of scientific studies. Each crossing creates a temporary disturbance. Each time, the halocline reforms. The pool persists. Patient. Exactly as it has been for centuries or millennia before any human vehicle descended to disturb it. The white mats at the edges of brine pools are not decoration. They are not mineral deposits or formations of sediment. They are alive, and they are among the most productive microbial communities on the deep seafloor. These bacterial mats are composed primarily of sulfur-oxidizing bacteria, organisms that take hydrogen sulfide, seeping upward from the sediment below, and use it as their energy source. The same way a plant uses sunlight, these bacteria use sulfur chemistry. They oxidize the hydrogen sulfide, extract the energy released by that reaction, and use it to build organic molecules from carbon dioxide. It is chemosynthesis, carried out on a quiet, continuous, vast scale. They are the base of the food web here. They grow slowly, spreading across the pale sediment in sheets and rosettes and tangled webs of microbial filaments. They do not move. They do not respond dramatically to disturbance. They simply grow. And in growing, they make a world possible for everything above them. If you drifted closer to the mat, you would see polychaete worms threading through the white fuzz, grazing as they move, small crustaceans picking their way along the surface, occasionally a fish hovering at the outer edge, drawn by the density of life, but careful not to approach the halocline too closely. The whole ecosystem is arranged around the boundary. The zone of maximum life is neither deep in the brine nor far out in the open ocean above. It is at the edge, at the gradient, where the chemical richness of the brine meets the oxygen availability of the overlying water. These bacterial mats also influence the chemistry of the surrounding seafloor. As sulfur-oxidizing bacteria process hydrogen sulfide, they produce sulfate. That sulfate dissolves into the surrounding water, changing the local chemistry. This influences which other reactions can occur in the sediment nearby, which minerals form, which other microorganisms can thrive. A brine pool and its mat communities are not isolated features on the seafloor. They are active participants in the chemistry of the surrounding environment, cycling sulfur between oxidized and reduced forms, removing hydrogen sulfide that would otherwise accumulate, connected to the larger ocean chemistry above them by the slow exchange of molecules across the halocline. Quiet. Essential. A part of how the ocean works. The connection between brine pools and the global carbon cycle is something researchers have only recently begun to map in detail. Methane is a potent greenhouse gas. In the atmosphere, it traps heat far more effectively than carbon dioxide, though it persists for a shorter time. The deep ocean floor contains enormous reservoirs of methane, stored in the form of methane hydrates in the sediment, and seeping upward through cold seep systems associated with brine pools and other deep sea features. If a significant portion of this methane were to reach the ocean surface and enter the atmosphere, it would have substantial effects on global climate. The microbial communities at brine pool edges, particularly the archaea performing anaerobic oxidation of methane, consume a significant fraction of this methane before it can rise through the water column. Estimates suggest that anaerobic oxidation of methane in marine sediments removes roughly 80 to 90% of the methane that would otherwise escape from the seafloor. This makes the microbial communities of brine pool edges and cold seep environments among the most important biological systems on Earth for regulating the composition of the atmosphere. A quiet, invisible service, performed in the dark by organisms most people have never heard of, in environments most people will never see. There is something worth dwelling on about the stillness of a brine pool. In the open ocean, water is never truly still. Currents flow through the deep basins of every ocean on Earth. The thermohaline circulation, sometimes called the Great Ocean Conveyor Belt, moves enormous volumes of water slowly around the planet, driven by differences in temperature and salinity. Cold, dense water sinks near the poles. Warmer, lighter water rises near the equator. The deep ocean turns over on a cycle that takes roughly 1,000 years to complete. Water that is at the bottom today was once at the surface. Surface water being cooled right now will eventually sink and spend a millennium in the deep before rising again. Even in the deepest and most remote basins, far from any storm or surface disturbance, there is movement. There is slow, patient circulation. Water is never entirely at rest. But brine pools sit outside this circulation. They are too dense to be lifted by ordinary deep currents. They cannot be stirred into the overlying ocean. They cannot be carried away. They simply sit. The brine in a pool may have been sitting there, in essentially the same place, for thousands of years. Possibly much longer. In the anoxic brine, almost nothing burrows into the sediment below. Almost nothing mixes the layers. Each layer of sediment is laid down and preserved in the order it arrived. By analyzing the chemistry of different sediment layers, researchers can reconstruct ancient ocean temperatures, ancient carbon cycling, ancient biological productivity in the surface waters above. They can identify periods of different circulation patterns, find evidence of ancient storms or floods, trace the record back tens of thousands of years to the last glacial maximum, when sea levels were lower and the coastlines of the world were different from today. The brine pool preserves all of this, layer by layer, century by century. And all of it has been sitting there, in the dark, since before most of the cities on Earth were built, since before the printing press, since before the earliest written records of most human civilizations, quietly holding its boundary, quietly hosting its microbial world, quietly being exactly what it is, in the deep dark, without anyone above knowing it was there. The ocean holds its secrets with extraordinary patience. The sediment beneath a brine pool also preserves something that is very difficult to find elsewhere. Intact pollen. Pollen is produced by land plants and carried by wind. Some of it lands in the ocean. In most parts of the ocean floor, pollen is rapidly degraded by microbial activity or mixed into sediment by burrowing organisms. But in the anoxic sediment beneath a brine pool, pollen can be preserved for tens of thousands of years. By identifying the pollen types in different sediment layers, paleobotanists can reconstruct what plants were growing on the surrounding land at different periods in the past. This means the sediment beneath a Red Sea brine pool can tell you what vegetation was growing across the Arabian Peninsula during the last ice age. The sediment beneath a Mediterranean brine pool can tell you about the forests and grasslands of ancient North Africa. These are records that simply do not exist elsewhere in the same quality. The combination of anoxic conditions, cold temperatures, and the extreme salinity of the overlying brine creates a preservation environment that rivals the best-known natural archives of ancient biological material anywhere on Earth. The brine pools of the deep are not just windows into the ocean's past. They are windows into the history of the land as well. As we come gently toward the end of our time at the brine pool tonight, let us think about what these places mean for the search for life beyond Earth. This is not a small question. Astrobiologists, scientists who study the possibility of life elsewhere in the universe, pay close attention to extreme environments on Earth. These environments, called extremophile habitats, show us the outer edges of the conditions under which life can survive and thrive. Each time an extreme habitat is found to support living organisms, the range of possible environments for life elsewhere expands. 2 moons of the outer solar system are of particular interest. Europa, a moon of Jupiter, is covered in a shell of water ice. Beneath that ice, evidence strongly suggests a global liquid water ocean in contact with a rocky seafloor, which may host hydrothermal vents driven by tidal heating from Jupiter's immense gravitational pull. An ocean where chemistry between water and rock has been occurring for billions of years, not entirely unlike the deep Earth oceans where brine pools and chemosynthetic communities have been found. Enceladus, a moon of Saturn, is smaller, but it is actively venting water vapor and organic molecules into space from cracks in its icy surface. It has a subsurface ocean. It has hydrothermal activity on its seafloor. It has the chemical ingredients that deep sea biology on Earth has shown to be sufficient for life, even in total darkness. The James Webb Space Telescope, launched in 2021, is capable of analyzing the atmospheric chemistry of planets orbiting other stars. Future missions to Europa and Enceladus are in various stages of planning. The Europa Clipper mission, scheduled to make multiple close flybys of Europa, carries instruments designed to study the chemistry of the moon's ice shell and any plumes venting from its interior. Each of these missions carries with it the knowledge gained from places like brine pools. The knowledge that life does not require sunlight. The knowledge that extreme salinity does not preclude life. The knowledge that the chemical energy available at the interfaces between different geological materials can support entire ecosystems in total darkness. The deep ocean floor is not just a place to study. It is a demonstration of what is possible when chemistry and liquid water are present, and time is long enough. The search for biosignatures, the chemical or physical signatures of life, is one of the most active areas of modern planetary science. A biosignature is any feature of a planet or moon that could only be explained by the presence of life. The most obvious would be a direct detection of living organisms, but biosignatures can also be chemical. An atmosphere with both oxygen and methane simultaneously, for example, would be a strong biosignature, because these two gases react with each other and would not coexist for long without a biological source constantly replenishing them. Some biosignatures are physical. Certain kinds of mineral formations, such as the stromatolites produced by ancient photosynthetic bacterial communities, can persist in the geological record long after the organisms that created them are gone. The study of brine pool ecosystems contributes to this search in a practical way. By understanding what chemical signatures the microbial communities of brine pools produce, researchers can better understand what kinds of signatures might be detectable from orbit around a distant moon. By understanding how life processes methane and sulfur compounds in the deep ocean, researchers can build better models of what atmospheric chemistry might look like above an ocean world with a similar biology. The deep brine pools of Earth are not a dead end in the search for life elsewhere. They are a training ground, a demonstration, a proof that life is more resourceful, more persistent, and more widely distributed than we once imagined. As we rise gently now, slowly back toward the surface, back toward the light, back toward the world you left behind when you closed your eyes, let the images we have visited settle softly into the quiet at the back of your mind, like sediment drifting back down through still water, slowly, without urgency, coming to rest. We began with the idea of a lake inside an ocean, something that should not exist, something the physics of water quietly insists upon when salt reaches the right concentration, in the right place, under the right pressure. We found ancient salt, buried for hundreds of millions of years under the seafloor, slowly dissolving into pools of impossible stillness. We visited the Gulf of Mexico and found communities of creatures thriving on chemistry alone, in the total absence of sunlight. We learned about the Alvin expedition of 1977 and how it shattered what science thought it knew about the requirements for life. We met the methane worm, living on ice made of gas, at the edge of a brine pool, in conditions that seemed designed to be uninhabitable. We sat with the Red Sea's hot brine lakes, heated by the slow separation of continents, hosting archaea from the oldest branches of life's tree. We hovered at the golden halocline, that impossible boundary where two kinds of water hold each other at arm's length, and found that the boundary itself was full of life. And we looked outward, to icy moons in the cold dark of the outer solar system, where oceans may wait beneath kilometers of ice, patient and still, perhaps not entirely empty. Through all of it, one idea stayed close. The strangest places are not the emptiest. The most extreme conditions are not the most lifeless. The edges are where things happen. The boundaries are where life gathers. And you, right now, lying still in the quiet dark of your own room, at the boundary between waking and sleep, are at an edge too. The place where the day dissolves into rest, where thought softens into something slower and quieter and deeper. There is nothing more you need to hold. Let the brine pools settle back into the dark. Let the halocline shimmer and fade. Let the bacterial mats drift gently out of sight. Let the deep ocean return to its own stillness, far below, going about its quiet business without you. And let yourself sink into the slow, unhurried passage of your own rest. The ocean floor is patient tonight. The universe is vast and old and does not require anything from you right now. And in this small corner of it, in this quiet room, pressed gently into your bed by the same gravity that holds the oceans to the earth, you are exactly where you are meant to be, resting, still, safe. If you are still with us, another quiet journey is waiting for you. And if this one helped carry you towards sleep, a like, a subscribe, or a gentle comment helps other tired minds find their way here too, from fall asleep, and from one tired mind to another, across the soft, patient dark of tonight. Sleep well, rest gently, and good night.